All right, so now I'm recording. So today's Thursday. I will be in the lab around 5 to about 7 today. I do have a meeting though at 4, and quite sure how long that's going to go. So hopefully I'll be able to make it to the lab right at 5. But if I'm a few minutes late, don't worry, I will be there. So just uh, hang in there. I should be able to get there pretty close to 5. So 5 o'clock uh, at the lab. And so hopefully that'll give anyone a chance to get in there and keep making progress. Also a reminder for as you are working through your lab project, taking pictures and documenting everything is important. I do expect to see pictures again, just similar to what you did with your field guide, because that can really help me figure out what's going on as you are describing your results. So take pictures of, plates or things under the microscope. Students have actually had quite good success taking pictures with their phone. Most students have better phones than I do because my phone doesn't take the greatest pictures, but you can do that. We also have a special microscope in the lab that has a camera attachment to it, and it's really quite straightforward to use. I could show anyone who wants to try that. I can show you how to use that as well. So a couple of different ways to take microscope pictures. But like I said, I would try your phone first because I've seen amazing pictures with just people taking a picture through the lenses that you look through on the microscope. Okay, so let's get into, whoops, all right. I'm gonna share my screen. All right, so hopefully you're just seeing one big screen. And I believe we left off right around here. Okay, so looking at the idea of, the microbe-human interactions, and when you think about the normal microbiota that live in and on us, how are we acquiring those? And I think if you just stop and think about it for a few minutes, some of these ideas would are kind of obvious. But when you think about the idea that, they're now discovering that the uterus actually does have some level of microbes present, and we're not talking about the pathogens, just normal microbes. And then of course, as the infant is going through the vaginal tract during the labor process and birth process, you will get plenty of colonization that way as well. There's even been a hypothesis that when you are undergoing the birthing process, and I've done that, so I have some familiarity with it. It is a messy process, and so they think that you get plenty of microbes. The child, the baby is getting exposed during that process. So the idea here is, again, we're talking about this vertical transmission of microbes from the mother to the infant, and then once the infant is born, it is, the baby's getting exposed to everyone else, not just the mom anymore. And so this idea that the baby is immediately exposed to the microbes in the vaginal tract, the feces as well. Like I said, birthing is a messy process, and so there is that exposure as well. And then just the skin-to-skin contact with the baby, with the caregivers, including the mom. And then if the baby is breastfed, you have microbes that are on the breast tissue, and they've actually done some really nice studies showing that microbes are also colonizing in the milk duct area of the breast, and that the breast milk can also have microbes in it, and beneficial microbes. And so that was a debate for a while too. They thought breast milk might be pretty much sterile. I mean, they knew skin, there would be microbes on the skin, but they weren't sure about the breast milk, but they're finding that there is some level of microbes present. And so, excuse me, another reason why, they're saying that if for feeding an infant, breast milk is best. And now they're showing that the beneficial microbes are also present there as well. And so that's another piece of data to support the idea that, we evolved to have breast milk. That's how our species evolved. Although, it's interesting, most mammals, you go through that weaning period where the youngster comes off of the breast milk. But yet you think about it, we're really the only species I'm aware of that continues to drink milk, but we drink milk from another species, which is a little odd when you think about it from the evolutionary perspective. So, of course, the milk industry doesn't want to hear me saying that, because that doesn't help the milk industry. But when you think about it, we're weaned off of our mom's breast milk within the first year or two, and yet we still drink another species. Is that just me? I think that's a little odd. I don't know. I think that's an interesting thing. And
I grew up on a farm with cows. They weren't dairy cows, but the mother cows obviously produce milk, and they all of the cows would drink it, not even just the babies. Like, they would drink their own milk. It was weird, just all of them.
Huh. Yeah, I've not been around dairy cows enough to know that. That's interesting. And I've drinking milk, humans drinking either cow milk or goat milk, camel milk too. I mean, it's people do it.
We had one goat on property, and it would even drink the cow milk.
So yeah, I mean, it's something that humans have done for probably thousands of years, and fermented milk too. That's been going on for quite some time. I still do fermented milk, yogurt and kefir, milk kefir and things like that. Sorry, my cat is going nuts right now, scratching on my furniture. So anyways, that's a whole nother discussion. I've done quite a bit of research into milk, and my own personal, I've backed off drinking milk. I still eat and drink fermented milk, but I just don't drink milk straight anymore for a variety of reasons, and that's a personal decision. So anyways, so I think you would probably quickly agree with your training in microbiology that, what you are exposed to on a daily basis, those microbes are what you're coming into contact with. And some of those microbes can be transient, where they get on and in you, but they don't persist. Maybe they're just there for hours or maybe a day or two, but then they get outcompeted. And then, of course, there are the ones that find a suitable niche either on or in us and are setting up camp and beginning to persist with us. And so certainly, if you think about, again, going back to the infant being born, all the caregivers and the family and the pets and the environment in the home, all of that comes into play. And at the time I was having children, I was doing a lot of reading, of course, about this idea. And I was actually nicely reassured that you shouldn't have a super clean house, and that, picking up all the dust bunnies and all the sweeping every day, it's not necessary. In fact, they say just the opposite. A little bit of dirt, so to speak, is good for exposure for children and babies. There was a New York Times article, and it had a very graphic picture of a baby sitting in a mud puddle with his fist up to his mouth, eating dirt. And the title said something like that, eating dirt, is that okay for your baby or something? And the gist of the article was, yes, of course you don't want to plop your baby down into a mud puddle and have them go at it. But they're saying that, you need to be exposed to a variety of microbes early on. So we'll get into that a little bit more. But then as they've done some nice studies where they track all the way from the birthing process to toddlerhood into adulthood. And they're gathering quite a bit of data, and they are showing that there is this succession of microbes as you grow up. And then as you get into your adult phase of your life, you do have this so-called adult microbial profile. And, you know, again, it's similar to the successional processes we've already discussed in this class and what you've been exposed to maybe in your intro ecology class, this idea that you will see a progression and a succession of microbes until you get more of a stable kind of climax community. And so what they're finding is when you start examining adults, and they've looked at, you know, adults from all kinds of different backgrounds, different geographic regions, and when you get into the adult profile, you tend to see these 4 phyla of bacteria dominating. So I do want you to memorize these 4 phyla because you're going to see them pop up over and over again. So the Bacteroidetes and the Firmicutes. So the Bacteroidetes are gram-negative, Firmicutes are gram-positive, Proteobacteria gram-negative, and then the Actinobacteria phylum is gram-positive. And so these 4 tend to dominate. So this is at the phylum level now. So that's pretty broad, right? Because, you know, you start with domain, and then kingdom, and then phylum, and then class, family, genus, species, or orders in there somewhere too. We're still talking pretty broad, all right? But know those 4 phylum, please, phyla. And then you know, it's not like we're all identical. That's not what they're saying, because like I said, this is at the phylum level. And so there's a lot of diversity within these 4 different phyla. And so you certainly, just like all humans are unique, you will see a unique profiles of certain types of microbes in one individual compared to the next. But if you rise up into the phylum level, you do see those 4 really dominating. So this is looking at the idea of the progression going from birth. So here they're looking at even the difference between if the baby is fed breastfed or formula fed. And again, they can see some major differences in what types of microbes initially colonize the infant, depending on what food they're eating, which, you know, if you think about it, it makes sense. That's the most intimate exposure you have to your environment, because not only are you exposed to it externally, but then you take it into your body, you ingest it. And so eating, your food choices are definitely the most intimate way you have of connecting with your external environment, because you actually bring it in to your body. And so here you can see some comparisons with the difference, and you do see a difference in the type of microbes at the phylum level here. All right. And then here, the middle column, looking at pre-toddler, infant to pre-toddler, as the toddler is being weaned and introduced to solid food. And you start to see an increase in diversity, of course, because there's more diverse food. It's not just breast milk or formula anymore. And then as you get into toddler, where there's more and more diverse food, of course, you start to see increased diversity. And, you know, the breastfeeding history ceases to impact because, you know, it's been too long now since the milk exposure has occurred. And so butyrate, we'll look at this idea, increasing butyrate bacteria. Butyrate, they're finding, is a pretty pivotal chemical. It's a product made through fermentation that bacteria do, and they're seeing some interesting impacts that has on our body, mostly good, but we'll look into this. And again, the Bacteroidetes and the Firmicutes showing up at that stage of toddlerhood. Okay, this is taken from that paper that we were just looking at, that figure. And 37 adults for up to 5 years, 60 to 70% of the strains present remained unchanged over the course of the study, and that the most stable members of the microbiome tended to be the most abundant. All right, so there's some nice you know, data that's showing there's a stable presence of microbes. At the phyla level, you have Bacteroidetes and Actinobacteria populations were less susceptible to perturbations. So that means they were more stable. They were more stable members of the community if there was any kind of difference in what the human was being exposed to. Whereas Firmicutes and Proteobacteria were significantly less stable. So what that's saying is if the human experiences a change in their environment or a change in their food choice, the Firmicutes and the proteobacteria, the microbes that fit into those two phyla, can get messed with a little bit more easily. And you can see shifts in the ratio of the organisms belonging to those phyla, the ratio and the population levels. So we'll get into that as well. And so here we're looking at this idea again of some really nice studies that are being published looking at the progression over time. So tracking, the same individuals in a long-term study. And you can see this is kind of nice just because you're kind of looking at this pie chart idea where you're looking at the different types of microbes. So they're doing it by phylum. So the Firmicutes, Bacteroidetes, Actinobacteria, and Proteobacteria, and then the gold is everything else. So, but you can see that you really do see, like here, I'm seeing a lot of Firmicutes as you get older, right? The pinkish part of the pie chart, so to speak. So the Firmicutes look to be starting to dominate as you get older. Bacteroidetes and Actinobacteria seem to be back when you're younger. All right, and Proteobacteria too, I guess, right? Oh, that's interesting. Malnutrition. And you see this big shift where you get a lot of proteobacteria and more Firmicutes. So we'll talk about that. So again, big picture idea here. What do we see? Well, those 4 phyla, again, they're tracking that and finding that those are the most abundant organisms, but you do see this successional progression of the microbes over time all the way to elderly state of our life cycle.
Would you say that most of the others in the 2 baby and toddler sections would be because they're usually constantly inoculating with like random different bacteria because they're putting it in things in their mouth?
Yeah, exactly. So yeah, toddlers are amazing in their ability to sample their environment, and kind of like if you've ever had a dog, a pet dog, similar idea. They constantly are sniffing around and picking up stuff and eating it, and so toddlers are kind of like that too. And that's why so many parents are like, oh, we have to have a super clean house. Oh my gosh. And it's like, yeah, true, you don't want a choking hazard on the floor. But actually, the studies are showing that you can be too clean in your environment, and it actually can set your child up for a world of hurt in terms of allergies and certain diseases. So rest assured, you can be a little lazy on cleaning like I am, and it won't be the end of the world. In fact, you might be doing some good. So anyways, so we've talked about this idea. I'm just bringing this up so you have this in your slide set. So we have the normal resident microflora that are pretty much with us. They're consistently there. And then I mentioned the transients. They're present for hours to months, but then they fade off. And then the opportunists are the ones that you have to be careful about because those are the ones that tend to cause problems. So typically when we say the word opportunist, it does have a negative connotation because they're taking advantage of certain situations where they can gain the upper hand and where they can get access to better nutrition and then start to reproduce to high levels, and that often can cause disease on our bodies. And so if you're immunocompromised, for example, whether you're on an anti-cancer drug treatment or immunosuppressive therapy, or even if you're just fighting off another disease, like if you have the flu, that makes you immunocompromised for other things to get in. And so this type of idea of, and also surgery or a major, like if you have a trauma to your body, if you get in a car accident and get wounded, for example, opportunists could end up in parts of your body that they normally never could reach before because you've been compromised in some way. And then they can potentially set up camp, so to speak, and start to live in that part of your body where they normally would never do that. And that can cause problems. So also taking antibiotics can also set you up this way for opportunists to take advantage. And I think I have mentioned that at least once or twice in the class, but we'll come back to this idea of what antibiotic production can do, especially to your gastrointestinal tract microbes. So this, just looking at the different parts of the body and where they're finding microbes. And again, when I took my first undergrad microbiology class, it was, they said that the womb was sterile. Well, now we know that's not true. They said the lungs were sterile if you were in a healthy state. That's not true either. There's actually microbes, certainly in the upper respiratory tract, but even in the upper lung tissue, they've done some sampling. Excuse me, and they have found normal microbes there, not the ones that are causing problems. And then, of course, even in your stomach, even with that low acidic pH, there are a handful of microbes that have adapted to that. We'll talk about that. And then, of course, millions and millions and millions of microbes in your intestinal tract, both the small intestine and the large intestine. So pretty much anywhere you can think of. Probably the bladder. I think they're still saying the bladder is sterile unless you have an infection. But I don't think they have found any beneficial microbes in the bladder yet. But I could be wrong. I just haven't searched for it lately. I haven't done a specific search.
Well, are there any in the kidneys?
I don't think so. But again, I haven't done a targeted search on that. So yeah, that's a good point. I forgot about the kidneys. Liver too, I don't know about that. So those sites, I would have to double check and see if they've actually searched for that recently. Certainly the urethra is colonized, and the vaginal tract as well, the rectum, of course. And obviously your skin is well colonized, but we'll get into different parts of the body. So getting at this idea of can you be too clean? And there was a video I wanted you to watch, but it was taken off of YouTube. I'm not quite sure why. It was a documentary that Nova put out, but it was just a short video clip. Anyways, I have another video clip that we can try to watch here. And it has to do with this. Originally, it was coined the hygiene hypothesis. And this was when I went to a microbiology conference. Boy, this is maybe 10 years ago, maybe a little bit longer than that. This idea was being thrown around a lot and gaining traction. And again, it's the idea of being too clean. If you have an ultra clean environment, that it can actually be harmful to, especially to babies being raised in that environment, because of course, when you're first born, your immune system isn't mature yet. It still takes time for your immune system to really mature fully. And so the idea here is that with the too clean of an environment, your baby isn't getting exposed to necessary challenges to kind of work out the immune system, make the immune system work and be able to kind of categorize and document, like, this is safe, this is safe, this is safe. Oh, ooh, no, this isn't safe. And so the immune system needs that kind of constant kind of challenge in order to fully mature in the right way. And so, there was this, it's getting better, but for quite a stretch of time, the companies that were trying to profit off of antibacterial products were doing huge commercial, marketing campaigns that you have to be, ultra clean, ultra clean. Oh my gosh, you're a terrible parent if you don't have a clean environment. And so they played on your fears of something could happen to your baby. And so you have to buy all these antibacterial products. They even made toys, plastic toys that were impregnated with antibacterial compounds, pacifiers that were impregnated with antibacterial compounds. And so it was kind of a crazy time where antibacterial products, not just soap, lots of other products too, were being marketed and used by many, many people. Well, we've already talked about this. That sets you up for selection pressure so that those microbes, if they're constantly being exposed to some kind of antibiotic substance, some kind of inhibitory substance, well, they're going to gain resistance to that. And sure enough, when the microbiologist went in and started screening for microbes and saying, are they resistant to these antibacterial products? And of course, no surprise to us, the answer is yes. And so it was really kind of a sad push And it was over a decade of this going on. It's gotten better. I think more people are more aware of this, and I hope you, as educated microbial students, you will tell other people around you that antibacterial products, you got to be really careful when to use them. I do use them occasionally. For example, if I had to change a poopy diaper and I had poop all over my hands, well, then yeah, I would use antibacterial products when I'm washing my hands. But that's about it. On my normal day-to-day, I would not do that. So this idea, and it's also, ever since the field of microbiology began, it began during the period where the microbiologists were super focused on germs in the negative sense, that looking at the microbes that can kill you. And so it's kind of interesting when you look at the history of the microbiology discipline, how just because of that, because it was set in that context of germs are bad, ooh, evil. And so that's why I think it's taken so long for us to recognize how beneficial microbes are, because there's just this very long history of, oh, germs are bad. There's these things that are invisible and they can kill you. Oh my gosh, and so that doesn't help the situation for sure. It's kind of created an obsession. And then a lot of, people that aren't educated about it, they just don't realize that, there's a lot of beneficial microbes out there. So I'm trying to get to the chat. I just saw someone chatted. And it's kind of, oh, okay. Thanks for that. So is that a book? It must be a book.
Yeah. Sorry, that's a book. It's about 400 pages, so it's some time, but it's all about it's like this new way of understanding allergies and autoimmune diseases, and it's really interesting. Like I read it probably 5-6 years ago when I didn't even really know anything about microbes, and it was still really interesting to me.
Okay, an epidemic of absence. I like the title.
Yeah, it's catchy.
Yeah. Oh, cool. Well, good. Thanks. That's something. 400 pages. I might have to wait till break, but yeah, that looks like a good one. So initially it was coined the hygiene hypothesis. Now they've switched and they're saying, that's probably not the best way to describe it. And so being more scientific, they've switched the term and now they're calling it the old friend hypothesis. So you'll see that in more current publications and the way people talk about it currently. They might reference the hygiene hypothesis, but more often I'm hearing people say the old friend hypothesis. And so this really started by a scientist who was very interested in looking at the absence of beneficial microbes in people with allergies, particularly asthma. And the rise in asthma in children has been astounding. If you've looked at that data, it is astounding how many more children have asthma in the current day and age compared to previous generations. And so that got scientists and immunologists going, what is going on here? And so they started looking for causes. And finally, the microbiologist got in there and said, hey, maybe it's because we're too clean and we're just sterilizing our homes and environments to the point where our immune system reacts inappropriately. So when you have allergies, it's basically your immune system misbehaving. It is oversensitive. It freaks out over something that it shouldn't be worried about, like pollen. Pollen isn't going to kill you. And yet if you're allergic to pollen, it's your body going, oh, pollen, oh my gosh, this is very dangerous. I have to mount a defense. And then you get the allergic symptoms. And so it's really your body just overreacting. And so it's not, well, asthma can be life-threatening, of course. Some allergies are life-threatening. But people do learn how to cope and live with certain drugs and ways of living their life to try to, knock down how often they have an allergic response. But it's very debilitating. It's not fun. And so, and then just the massive increase in the amount of children, especially, that were getting asthma and other allergies, really made the scientists say, we got to look into this. There's got to be something going on here. And they did look at a range of different pollutants. They were targeting all kinds of pollutants that are in our environment now due to a variety of different industrial practices. And then they also started looking at the absence of microbes in the environment that the child is raised in. So that
Did they
came.
look at
Go ahead.
interactions between the pollutants and the beneficial microbes? Like if one pollutant was specifically like killing off a specific beneficial microbe?
I don't know if they looked at it in that manner. I think when they were looking at pollutants, they were looking at directly how does the human body respond to these pollutants, and not so much looking at the impact on the microbes. And so they were worried that the toxins in the environment, whether it was something in the drinking water or something in the food or something in the soil that the child was exposed to, or in the air, of course, what you breathe in, they were looking at the direct impact on the human body. As far as I know, at least the readings I read. So, so I put on this slide, watch the Germany study. That's the one I couldn't find. I had it posted in EduCap, but the link is gone. They won't let me into it. So I found something else that's similar, so we'll watch that in a second. But I thought this was a nice throwback to Charlie Brown. That's Pigpen, right? That's his name? Yeah. Pigpen, always dirty. And anytime he'd walk, you'd see the clouds of dirt and all of that coming off of him. So again, just getting at this idea that you can be too clean. But let me see if I can pull up that little short video. It's just a couple minutes long. Let me see if I can stop share maybe and get into it. Hang on now. Okay. Let me. Okay, let me share again. Whoops.
Can you tell us a little bit about the hygiene hypothesis?
Are you seeing this sexual movie? Are you
Yes.
seeing it? Okay. So this is just a couple of minutes long.
Sure. This was a hypothesis that was put forward by somebody called David Strachan in 1989. And basically, he came up with this idea to try and explain why we saw so many more people getting allergies in the 20th century.
What do you think is the role of the world around us in changing how our immune system works?
So what Strachan noticed was that in the country, in particular farms, people tended to have less allergies than people who lived in cities or towns. He also noticed that people who were in very large family groups, so a lot of brothers and sisters, were also less likely to have allergies. So he wondered if it was early exposure to germs that was really protecting people from allergy, and you were more likely to get that exposure if you had a big family or you were living in the country.
So what are some possible explanations for this?
Well, the idea would really be that the early exposure to germs is educating your immune system and giving you a fit immune system so that it doesn't start responding incorrectly to things that should be harmless. Because basically what an allergy is you reacting to something that's completely harmless that you should ignore.
So is it all germs that we should be thinking about here, or are there some specific ones involved?
So that's a very good question. There's been a lot of research on this, looking at all sorts of different bacterial infections and also viral infections. And the research has basically produced very conflicting results. They're just not clear. However, the hypothesis has moved on to focus on the old friends hypothesis. And that introduces the idea of parasitic worm infections and their role in perhaps protecting us against allergy.
So your own work is looking at parasitic worm infections. Can you tell us a little bit about that?
Yeah, we are very interested to understand how your immune system deals with parasite worm infections. And in particular, we're trying to understand the products that parasites make and how they manipulate the immune response to promote their own survival. And lots of other groups are doing that as well all over the world. There's even clinical trials using parasites to try and prevent disease. The other thing that we're trying to do is we're trying to understand why some people have a very bad immune response to worms, because obviously you don't want to give somebody like that a worm infection. So we're looking at biomarkers that will help predict the people who are less likely to do well with worms.
Okay. So, an interesting idea. Let me go back to, whoops. No, stop share. So the old friends hypothesis that she mentioned, this is the idea of the if you think about our evolution, you know, the human species over evolutionary time, and that, come on now, that we have had plenty of exposure in our past as well as our current lives with parasites, with worms. And anyone who's taken parasitology, which is a fun class, I took that when I was an undergrad, very fun. If you're a microbiology major, that's another one to consider. You know, it's not just other animals that get parasitic worms. We do as well, although it's, you know, we have generally, you know, most physicians in the current day and age think that's a bad thing, and they will try to immediately give you some kind of treatment to get rid of them. Well, there's a group of scientists that are saying, well, wait a minute, you know, if you think about the evolution of our species, of humans over time, we've interacted with parasitic worms for thousands of years. And maybe we should reconsider, in the light of that information and that knowledge, maybe we should reconsider going on an all-out war against any little parasitic worm that might be inhabiting us. You know, is that necessarily an awful bad thing? And so, of course, this has been kind of a slow process of acceptance to kind of open our minds to this idea. And especially, I would imagine physicians would have a hard time with this. And so, I mean, I don't know, what about you? The idea of carrying worms inside of you on a regular basis as part of your microbiome, I guess, it's kind of an odd thought, right? Although, apparently, I don't know if it's still a thing, but I heard maybe 10, 15 years ago, supermodels were deliberately swallowing tapeworm eggs. Have you heard this?
I've heard that there's like diet pills that have tapeworm eggs in them.
Oh my gosh. So apparently, you know, if you're a supermodel and that's how you're earning your money, you have to stay a certain weight, and you cannot vary, you know, you can't increase your weight or you won't get the job. So yeah, I heard that that was a fad for a while, and probably not just supermodels, probably some people who are super fanatic about their weight, they would swallow tapeworm eggs. And if you have taken parasitology, you might have seen, or with your own eyes, how tapeworms can inhabit the intestinal tract, latch on to the lining of your intestinal tract, and siphon away nutrients from your body, and then reproduce themselves. You know, lots of egg production, and then the actual tapeworm itself can get quite large and be a rather large member of your community, of your body. So you have to be pretty fanatic, I guess, about weight to do something like that. In my opinion, I cannot imagine. But anyways, having said that, you know, this idea of parasites and worms in particular. So that conference I went to, where they were talking about the hygiene hypothesis, there was a scientist from over in Europe, I'm forgetting which country. He was studying populations in Africa, human populations, and looking at the correlation between parasitic infections in that community and asthma rates. And he was showing some really nice data. Again, you know, correlation is not causation, so you have to be careful. And these were some of his initial studies. And like I said, that was many years ago. And they've made progress now where they're more confident in saying that there seems to be something here with not just the microbiome, the bacteria that are inhabiting us, but also any of the parasitic worms. And so I think you're going to hear more about that in your life, and this idea of having parasites in your body may not be as evil and bad as we thought. So thinking about our gastrointestinal tract, let's hone in on that a little bit. Literally trillions of bacteria. And don't forget about viruses too. They're starting now to look at the viral biome of humans as well, which is a little bit more challenging, but they're finding that we have lots and lots of viruses, like the bacteriophages, that, remember now, bacteriophages infect bacteria, but they don't infect human cells. And so they are pointing to the idea that there's plenty of microbes in our bodies that have bacteriophages in them, and that can be one way to keep population levels in check, because if they lyse the bacteria, of course, they're killing the bacteria. And so just like in regular ecosystems, they're finding that viruses are a way to kind of check the population of microbes so that they don't get overwhelming high populations in different habitats. But anyways, if you start looking at our gastrointestinal tract, and if you start from the stomach and work your way forward from there, initially, again, they thought, what could survive in stomach area, because the pH is so incredibly low, pH 2 to 3, I think, or in that range. And so the famous example is Helicobacter pylori, and that microbe is very well adapted. And again, this kind of goes along with the old friends hypothesis. There's another group of scientists who are saying Helicobacter should not be so immediately thought of as evil, and we have to, pop antibiotics and get rid of it. And so it's been an interesting, I guess, debate in this idea, because some of you may already know, I might have mentioned it too, maybe, that Helicobacter is now, it's been shown quite firmly that it is the microorganism that can cause ulcers in your stomach. And initially, this was not ulcers. They thought you got it from spicy food. They said if you eat really spicy food and if you are under a lot of stress, you might get an ulcer. But nowhere were they thinking about a microbe doing this, because they just didn't think microbes could handle the stomach. It was too acidic. And then two scientists, I believe they're from Australia, they were looking at this and they said, I bet it's microbial. And they started looking for it, discovered Helicobacter pylori living in the, on and kind of embedded in the lining of the stomach. And they were convinced, but nobody else was. And so at a national conference, what they did was they grew up Helicobacter pylori, and then on the stage, as he's giving his presentation, he swallowed the vial of Helicobacter and said, I'll prove it that this bacteria causes ulcers. And so sure enough, he got an ulcer. And luckily, antibiotics can help rid you of that if you have a really bad ulcer. And so very graphically, I mean, that to me is a little extreme passion for your science. I mean, I really like science too, and I really like microbes, but I don't think I would stand up on stage and swallow one and say, okay, watch me get an ulcer. Anyways, go ahead.
They use antibiotics. Were they using antibiotics to cure ulcers before they knew this?
I think some doctors were doing it, but they didn't quite link it. They still were having a hard time believing anything could live in the stomach. When my dad was probably in his 50s or 60s, I remember him complaining that his stomach was hurting, and he went to the doctor and they said, yeah, you have an ulcer. And they said, stop eating spicy food. And when my dad came home and told us all that, we laughed so hard, because my dad was 100% Finnish. If you know anything about the Finnish cuisine, Finlanders do not eat spicy food. That is not what you do. And I grew up on meat and potatoes and very bland food in general. So we kind of laughed at that. So he was under a lot of stress, and I think the stress was weakening his immune system to the point where Helicobacter started to overgrow a little bit and cause problems. But I don't think he got prescribed antibiotics back then. So, but yeah, you can take antibiotics to kill off Helicobacter, and it can work. But again, there are some scientists saying, you know, just because you have Helicobacter, by the way, you know, that doesn't immediately mean you're going to get an ulcer. It just means you have Helicobacter. It's more of if you create a situation where you have dysbiosis, an unbalanced state in your body where Helicobacter can gain the upper hand in terms of ramping up its population, then it can start to cause some problems. But just because you have Helicobacter does not mean you're going to get an ulcer. So be careful about that. So Helicobacter is one of the major ones that they've discovered. There are a couple of other species that can handle the very acidic pH. Now I won't go into huge detail, but basically what they're doing is they embed themselves a little bit into the stomach lining, and then they excrete molecules that locally raise the pH around them. So they have a way to kind of create a little home environment where the pH is not quite so acidic, and then they're happy. They can survive that, and they have a very non-competitive niche because there's so few other microbes that can handle that. So, and then as you work your way past the stomach and get into the small intestine and the large intestine, then you start to see much more, many more species, much more higher populations of microbes showing up in these parts of the gastrointestinal tract. So we're going to start to go through this as we work our way into this topic. The large intestine in particular has a high abundance of obligate anaerobes, which is kind of weird. You're like, well, wait a minute. I mean, I breathe in air, there's air in my body. How can there be anaerobes? But you have to remember, again, the size difference. Microbes, you know, just 3 micrometers in size roughly. And so it's very easy for them to find anaerobic environments in your intestinal tract. It's not that hard. And massive population levels, 10 up to 1 times 10 to the 11th CFUs per gram of intestinal tract tissue. So lots and lots of microbes. And we're going to get into this. And then, of course, there's facultative anaerobes. And that can be also, but they tend to be at a lower level. The obligate anaerobes tend to dominate in the large intestine. And so when they look at fecal matter and estimate how much of the feces is food that we didn't digest and how much is microbes, it's about 50%. Some reports I've read it's higher than 50%, but I guess a good number is roughly 50%. You can think of feces by weight and volume is bacteria. So there's plenty there. And so I think what I'd like to do is kind of work through the different parts of the GI tract. I did post into the EduCat, there is a reading on comparing the intestinal tract all the way from actually probably the mouth all the way to the rectum, and comparing it to stream ecology. And so I thought that for those of you who might have taken stream ecology or even intro ecology, I thought it's a nice comparison between what's happening in a stream system versus what you can think of happening in the GI tract. And so please read that. And then I've also posted a couple of other readings that we'll get into on the hologenome theory, which you can hold off on those a little bit if you want, but the GI tract one and stream ecology, I'd like you to look at that one. And we'll get into that next as we continue with this topic. Okay, so tomorrow, and remember tonight at 5-ish, I will be at the lab to have an open lab for anyone who wants to come in. Tomorrow's lab from 10 to noon. Again, anyone can come in, and hopefully everyone who arrives can fit in, and we'll be able to make more progress on the research. Are there any questions?